I would like to thank Dr. Chip Chambers and the organizing committee for the invitation to present at this meeting. And here is the proposed title of my talk today. Briefly to remind you, deficiency of ADA2 or DADA2 is recessively inherited disease caused by either hypomorphic or loss of function variants, pathogenic variants in the ADA2 gene. Typically, these variants are missense. Most patients have missense loss of function mutations in this gene. However, there is increasing number of patients that are identified to carry nonsense, frame shift, or copy number variations, and copy number variations are the topic of my presentation today. Pathogenic variants are either novel or present at a low allele frequency or are found at a low allele frequency in the general population. And we also have to think about population-specific variants. And these are basically the staples that we have to think about in molecular diagnosis of deficiency of ADA2. Patients that have DADA2, if they come from the outbred population, typically present as sporadic cases in the families. However, there are some families that can have more than one affected child. DADA2 is also common in many founder population with consanguineous family, as is the case with this family of Turkish ancestry with multiple inbreeding loops and the multiple basically affected cousins in the last generation. In addition, a recent study from India basically reported that 16 out of 22 patients that were molecularly diagnosed with DADA2 were homozygous for respective mutations. In particular, 14 of them were homozygous for this missense mutation, glycine 47 to arginine. This particular mutation is associated with vasculitis or polyarteritis nodosa, as it was shown in this paper by Dr. Pyu Li and Ching Zhou. And this variant has a high carrier frequency in multiple founder populations, starting with the carrier frequency of 1 in 10 in the Georgian Jewish population, 1 in 500 in the Turkish population. I estimated that the carrier frequency of this variant is about 1 in 700 in South Asian population based on the data from the GNOMA database. Interestingly, this variant is not found in any of the 20,000 people of East Asian ancestry that are tested in the GNOMAD database, and carrier frequency of this variant is also much lower in the European and Latino population. Before I go further, I would like to remind you also that there is evidence for reduced penetrancy in patients with DADA2, as is the case with this family of Pakistani ancestry with 4 family members who were homozygous for this particular missense hypomorphic mutations in the gene, and basically also had low ADA2 levels. However, they didn't present with any clinical symptoms, in contrast to 2 affected family members that presented with the typical DADA2 features. Basically, this suggests the role for other modifying genetic alleles, epigenetic, and other environmental factors in the disease expressivity of DADA2. ADA2 gene is highly polymorphic, and there are close to 400 variants that are identified in the coding region of this gene, as it's shown here in this table that I found again in the GNOMAD database. So DADA2 associated variants are either novel or they are found at a lower frequency in the general population, and that frequency can be as high as 1 in 1,000. We also take advantage or use basically different algorithms or these in silico prediction tools that basically support further that these variants have a damaging effect on the protein function when working on diagnosis of patients with suspected DADA2. Here is the figure from the paper that was written 2 years ago, and I can tell you that this figure is already outdated, but I just didn't bother to update the figure with novel mutations. But basically the point of this figure is really to show that these pathogenic variants reside or are identified in all protein domains. I then went to the InFevers database, where basically you can sort of generate a new figure as these mutations are updated in this database. And basically what I'm showing here is that the pathogenic variants are identified in every exon of this gene, including the first exon, which is untranslated. Essentially, it means when we do molecular diagnostic for DADA2, we have to screen the entire gene. And fortunately, this gene is not really big, so it's not a lot of work to do the screening. So now I would like to talk about hidden mutations or cryptic mutations that can also lead to deficiency of ADA2 and how to identify them. So here is the family that was referred to us several years ago, and the problem in this family presented with clinical manifestations that were consistent with the other two, namely with livedoid skin rash and also with multiple strokes. The other three siblings were unaffected at that time. So what we did in this family is just standard Sanger sequencing and basically showed that the proband inherited this known DADA2 associated missense mutation from the father, and to our surprise, this mutation was also identified in the other three siblings. We then measured the ADA2 activity in the family and again showed that both proband and also the two unaffected siblings in this family had absent ADA2 activity, which essentially means that these two siblings have DADA2. The question is how to find the second mutation in this gene. So we sequenced cDNA samples of these patients and basically showed that the second allele is not expressed, as shown here in this electropherogram, where you can see that in a patient that does not have the second mutant allele, or basically in this sibling, you see double clicks, which is not the case with other three siblings that have DADA2. We then did haplotype analysis in this family and basically showed that the mother is the carrier for mutation, which is here shown or depicted with green color, and that this mutation is passed to all three DADA2 siblings, or all three siblings with DADA2, whether they are affected or not affected. So finally, we did whole genome sequencing in this family and identified the missense variant in the 5' UTR region of this gene, and this missense variant basically leads to the reduced expression of the ADA2 transcript, as shown here in this figure, where you can see that in the mother, which is shown with the white bar, and the other three siblings that carry this mutation, the expression of ADA2 is at about 50% compared to healthy controls. Well, basically, we confirmed by doing all these assays that three siblings in this family are compound heterozygous for two different pathogenic variants in this gene. At that time, when we made this clinical or molecular diagnosis, clinically, these two siblings were still unaffected. Unfortunately, the male sibling in this family, maybe about a year later, presented with a minor stroke, and of whom he recovered eventually, and since then has been treated with TNF inhibitors. So now I would like to talk about copy number variations or structural variants that can also lead to deficiency of ADA2. So what are copy number variations? Basically, they are DNA segments that are present at the variable copy number in genome. They can lead to gain or loss of genetic material, but they are not always pathogenic. So here shown is the ADA2 genomic region, and here on the top of this panel you can see the structure of different isoforms of the ADA2 gene, and exons are here shown with these dark boxes. But what I would like you to pay attention to are all these lines, shorter lines, either colored with blue or red, which essentially show that there are a lot of small copy number variations within this gene or around this gene. With the help of the bioinformatics student in our lab, Brunja Matthias Sardotir, we made this figure, and basically this figure showed again that ADA2 genome or gene region is rich with copy number variants, and there are many more copy number variants in this gene than, for example, in other genes that are associated with different monogenic autoinflammatory diseases. In particular, I would like to talk about one copy number variant that involves exon 7 of this gene, which essentially leads to, when duplicated or deleted, lead to a shift in protein translation. We estimated that the size of this copy number variant is about 800 base pair, and in patients that have DADA2, basically they are either homozygous for the duplication or the deletion of this copy number variation. In addition, this copy number variation can be also inherited in combination with a second pathogenic variant, typically missense mutation in this gene. The potential underlying mechanism for the pathogenic event is a non-homologous meiotic recombination event between two repetitive allo elements that are flanking exon 7. These two allo elements are highly homologous, and they basically have about 86% of the identity. in their sequence. So here is the family that was diagnosed many, many years ago, as it says in the title, with PAN, polyarteritis nodosa at Johns Hopkins. So basically, we sequenced these 3 affected siblings and showed that they carry this missense mutation pathogenic variant in the ADA2 gene. The question was again, where was the second mutation? We did cDNA sequencing, which showed that the second allele again is not expressed, as shown here by the loss of this double peak in the electropherogram. ADA2 activity assays showed that these 3 siblings have very low ADA2 activity, which essentially confirmed that they have DADA2. So the next step was to do search for copy number variation in this family. At that time, actually, we developed finally this assay, MLPA assay, which stands for multiple ligation probe assay, that we use nowadays to screen for copy number variations. And by using this assay, we showed that all 3 of them are carriers for the deletion of exon 7, which essentially means that these 3 affected siblings presenting with PAN are compound heterozygous for 1 missense mutation in the gene and exon 7 deletion. Here is another family with unusual inheritance pattern. This family is of Pakistani ancestry, and basically in this family there was a proband which presented with congenital anemia soon after birth and basically required frequent blood transfusion starting at the age of 3 months. This proband didn't have any history of episodes or any significant infection history. Because this proband was initially diagnosed with DBA or Diamond-Blackfan anemia, she was screened for mutations in genes that are associated with DBA, and after she tested negative, basically she was screened for mutations in the ADA2 gene. So basically, we failed to identify pathogenic variants by doing just regular sequencing. And the next step was to do ADA2 activity assay, which essentially showed that the proband, but also to our surprise, her father, who is clinically asymptomatic, and another sibling who is young but clinically asymptomatic, basically had absent ADA2 activity. Essentially, that confirmed, again, this functional test confirmed that they have a deficiency of ADA2. Using MLP assay, we showed that basically these 3 family members are homozygous for the duplication of exon 7, or that they have 4 copies of this copy number variation. Father is still asymptomatic, except that he has hypogammaglobulinemia, and another sibling, I believe she is still asymptomatic, and I know that she also was diagnosed with hypogammaglobulinemia, and I think both of them also had a higher autoantibodies, anti-double-stranded DNA autoantibodies. So again, to our surprise, in this family, we identified 2 family members that are asymptomatic, so there is evidence for reduced penetrance. But in addition, when you look at this family, you would think that this is a dominantly inherited trait, but actually it's a recessive disease. And due to the fact that this mutation is, I shouldn't say common, but it's present in that inbred family, there are some families that will present with a disease that it appears as a dominant inherited trait. This is not the only disease where you can see pseudodominance. For example, we've seen examples of pseudodominance in families with FMF. So because in some countries or in some population, it's not really doable to screen for copy number variations, we proposed another way another test that can be used to identify copy number variations based on the studies that we did in the families discussed in the previous slide. So what we did basically is that we developed long-range PCR primer set flanking ADA2 exon 7, and what you can see here in this figure is that the father and the 2 siblings who have DADA2, whether they are affected or not, basically have this higher molecular band, which represent homozygous duplication of exon 7. This is again compared to the healthy control sample and to the mother who is the carrier for this exon 7 duplication. Another way to test for the possible copy number variations or hidden variants in the gene would be also to use QRT-PCR assay, which can demonstrate a low expression of the ADA2 transcript, as this was the case with this family. Searching the literature, I found this paper, which basically described another patient of Italian descent with DADA2, typical clinical manifestations, and basically absent ADA2 enzymatic activity. Interestingly, this activity was measured in proband monocytes, which is not what we typically do. Typically, we measure the ADA2 activity in peripheral blood samples, either plasma or serum samples, and they use whole genome sequencing to basically show that this patient is the homozygous for tandem duplication of the genomic region encompassing exons 3 and 4 of the ADA2 gene, resulting in a frame shift and premature stop codon. I would also like to say a couple of words about these copy number variations that I've heard in the past year in several actually patients that don't really present with DADA2, but nevertheless they were identified to carry this exon 8 to 10 copy number variations. In particular, I heard about the family with 2 siblings. Both of them were heterozygous for duplication of exon 8 to 10, which essentially means that they had 3 copies of these copy number variations. ADA2 activity was measured in one of these siblings, affected siblings, in Dr. Hirschfield's lab, and basically was shown to be normal, which essentially suggests that this copy number variation is unlikely to be pathogenic. In addition, these 2 siblings presented with features that are not really suggestive of DADA2, namely adult onset recurrent fever and pericarditis, and we believe that there is another gene causing this phenotype in the family, so we plan to do whole exome sequencing in the family. So finally, I would like just to show you the proposed scheme for molecular diagnostic testing in patients suspected to have DADA2. So the first step we do is actually Sanger sequencing, or you can do targeted gene panel sequencing, whole exome sequencing, whatever is the fastest way to do the sequencing. Of course, one can circumvent this step and just go straight and do ADA2 functional assay, which is also confirmatory of DADA2. So if we identified 2 non-pathogenic variants, that is basically confirmatory of DADA2. If in some patient only 1 pathogenic variant is identified and the second variant is novel, then we go for the ADA2 activity test, which can or should confirm the pathogenicity of this novel variant. Another thing to do would be to do segregation analysis in the family. In patients in whom we identified 1 pathogenic variant and the second variant is not identified, again, we propose to do ADA2 activity test, and if this test is positive, or if this patient has a low ADA2 activity, then basically we should search for copy number variations in this patient. In patients that have 2 novel variants identified in this gene, again, the next step would be to measure ADA2 activity, and basically that would confirm the pathogenicity of these novel variants. In patients in whom sequencing does not identify any pathogenic variants, as it was the case with the family that I previously discussed, the next step would be to do ADA2 activity test if the phenotype is consistent with DADA2. If again now this ADA2 activity is low, then the next step would be to search for copy number variations either by MLPA or by targeted deep resequencing, whole exome sequencing, or whole genome sequencing, depending what is available. And this is the end of my presentation, and I would like to thank you all for your attention.